Somewhere in Europe, more than 30,000 years ago, one single human being carried a tiny copy error in their DNA. Just one letter out of billions changed by accident. That person had no idea what they were carrying. They lived, they hunted, they raised children, and they died like every other person of that time. But before they died, they passed that one changed letter to their children, and their children passed it to theirs, generation after generation, through ice ages, migrations, wars, and famines. That single genetic accident kept traveling forward in time, hidden inside the blood of millions of people who never knew it was there. Today, scientists estimate that more than 20 million people alive right now carry that exact same mutation. Not a similar mutation, not a copy that appeared again somewhere else. The exact same one, inherited from that same person who lived 30,000 years ago. This is one of the strangest and most personal stories DNA has ever told us, because it is not about a whole population or a whole continent. It is about one person, and how one tiny accident inside their body is still shaping human health today. If this kind of story interests you, hit subscribe, because every week I break down discoveries like this, the ones hiding inside your own DNA that connect you to people you will never meet. Now let's get into how scientists found this, and why it matters more than you might think. The story starts with something that seems completely unrelated to ancient history: blood clotting. Every time you cut your finger, your body performs an incredibly fast chemical chain reaction. Proteins in your blood activate one after another, like a row of dominoes falling, until a clot forms and the bleeding stops. This system has to work perfectly. If it is too weak, small cuts turn into dangerous bleeding. If it is too strong, blood clots form inside your veins when they should not, and those clots can travel to your lungs or your brain and kill you. One of the most important proteins in that clotting chain is called factor V. Normally, once a clot has done its job and stopped the bleeding, another protein called protein C steps in and breaks factor V down, switching the clotting system back off. It is a built-in safety mechanism. Turn clotting on when needed, turn it off when the job is done. But in the 1990s, doctors in the Netherlands were trying to understand why some of their patients kept developing dangerous blood clots for no clear reason. These were not people with obvious risk factors. They were not elderly, not smokers, not sitting still for long periods, yet their blood kept clotting when it should not have. A researcher named Rogier Bertina led a study to look closely at the DNA of these patients, searching for anything unusual in the genes responsible for clotting. What they found was a single change in the gene that builds factor V. Just one tiny alteration at one specific spot in the gene out of the 3 billion letters that make up human DNA. That one change made factor V resistant to being shut off by protein C. In simple terms, the off switch stopped working properly. The clotting protein stayed active longer than it was supposed to, which meant blood was more likely to clot when it should not. The mutation was named factor V Leiden, after the city in the Netherlands where the discovery was made. At first, it seemed like a fairly ordinary genetic finding, one more piece added to the long list of mutations linked to disease. But then researchers started testing wider and wider groups of people, and the numbers coming back were strange. This was not a rare mutation showing up in a handful of unlucky families. It was showing up again and again, in patient after patient, across country after country in Europe. Roughly 1 in 20 people of European background carry at least one copy of this mutation. In some regions, especially parts of Northern Europe, the number climbs even higher, closer to 1 in 10. To put that in perspective, that is not a rare genetic accident. That is one of the most common inherited clotting mutations ever identified in humans. Something that common does not usually stay common by pure chance. Mutations are supposed to be rare, unpredictable, and scattered randomly through a population, like static noise. When scientists see a mutation this common, concentrated this heavily in one part of the world, it raises a question. Where did it actually come from, and why did it spread so far? To answer that, researchers turn to a technique that lets you read the genetic material around a mutation almost like reading the growth rings of a tree. Every time DNA gets copied from parent to child, tiny errors and reshufflings happen nearby the mutation, a process called recombination. The longer a mutation has existed and been passed down through generations, the more scrambled and varied the DNA sequence around it becomes, because each new generation adds a little more mixing. But if a mutation appeared recently, the DNA surrounding it will still look almost identical in everyone who carries it, because there has not been enough time for it to get shuffled apart. When scientists studied the DNA sequences surrounding the factor V Leiden mutation in thousands of unrelated carriers from different countries, they found something remarkable. The surrounding genetic sequence was almost the same in every single person tested. Whether they were from the Netherlands, Sweden, Spain, or Britain, this meant one thing. All of these carriers, spread across an entire continent, thousands of kilometers apart, speaking different languages, living completely separate lives, had all inherited this mutation from the exact same original ancestor. Using the amount of genetic mixing that had occurred around the mutation, scientists were able to calculate roughly how long ago that ancestor lived. The estimates pointed to somewhere between 20,000 and 30,000 years in the past. That places the original carrier of this mutation deep in the last ice age, long before farming existed, long before villages, long before anything we would recognize as civilization. At that time, small groups of hunter-gatherers were surviving in a Europe covered in ice sheets, mammoths, and vast frozen tundra. Somewhere among those small, scattered bands of humans, one person was born with this tiny change in their factor V gene. This raises an obvious question. If this mutation makes blood clot too easily, and dangerous blood clots can be deadly, how did it not just disappear? Evolution tends to remove harmful mutations over time, especially ones linked to a higher risk of death. A mutation that increases the chance of a fatal blood clot should, in theory, get weeded out of a population across thousands of years. Instead, this one somehow became one of the most common inherited mutations in an entire region of the world. Something must have been protecting it, or even pushing it to spread. Researchers believe the answer lies in a completely different and much older part of human life, one that shaped survival for almost the entire history of our species: childbirth. For most of human history, giving birth was one of the most dangerous events a person could survive. Severe bleeding during and after childbirth was one of the leading causes of death for young women. Before hospitals, before blood transfusions, before modern medicine of any kind, a woman who bled too heavily after delivering a baby had a very real chance of dying within hours. A mutation that makes blood clot slightly more easily and slightly more strongly is not obviously helpful in modern life, where the bigger danger tends to come from unwanted clots later in adulthood. But in the world of the Ice Age, where childbirth bleeding was a constant and immediate threat to survival, a genetic advantage that helped stop bleeding faster after childbirth could make the difference between life and death right in that moment. A slightly stronger clotting response was not a liability then. It was potentially life-saving. This means the very same mutation that today gets flagged in a modern blood test as a clotting risk may have spent thousands of years acting as a survival advantage, helping mothers live through childbirth long enough to raise the children they had just given birth to. Natural selection does not judge a trait by whether it is convenient for someone living in the 21st century. It judges a trait by whether it helps a person survive and reproduce in the conditions they are actually living in. And for tens of thousands of years, in an environment full of danger during birth and few options if bleeding started, this mutation may have quietly given carriers a real edge. If that theory is correct, it would explain why the mutation did not fade away over the generations the way most mutations eventually do. Instead of being erased by natural selection, it may have been actively favored by it. Mothers carrying the mutation may have survived childbirth at higher rates than mothers without it. Those surviving mothers passed the mutation on to their own children, who then had daughters of their own, some of whom also carried the mutation and also survived childbirth more often. Over tens of thousands of years across hundreds of generations, this small advantage compounded, spreading the mutation further and further through the population, until it became one of the most common inherited traits in an entire region of the world. This is the same evolutionary logic that explains other mutations linked to disease today. A genetic change that provides a clear survival benefit under one set of conditions can persist and spread through a population even if it carries a cost under different conditions, especially if that cost mainly shows up later in life, after a person has already had children. Nature does not optimize for a long, comfortable old age. It optimizes for surviving long enough to reproduce and raise the next generation. A mutation that helps you survive childbirth at 20, but slightly raises your risk of a blood clot at 60 is, from evolution's point of view, still a winning trade. There's another layer to this story that connects it to the wider map of human migration. When scientists mapped how common the factor V Leiden mutation is across different regions of the world, a clear pattern emerged. It is most common in Northern and Western Europe. It becomes less common as you move towards Southern Europe and the Middle East. It becomes rare in parts of Asia, and in populations whose ancestors never passed through Europe at all, such as many indigenous groups in East Asia, Sub-Saharan Africa, and the Americas, the mutation is almost completely absent. This pattern lines up closely with the story of how humans spread across the planet after leaving Africa. Small groups of early humans moved out of Africa tens of thousands of years ago and gradually spread into the Middle East, then into Europe and Asia, then eventually across into the Americas via the far north. If a mutation appears after a population has already split off and settled into a specific region, that mutation will usually stay concentrated in that region, and in the descendants who later migrate outward from it. It does not automatically appear in populations that split away earlier and never had contact with the group carrying it, because the factor V Leiden mutation is heavily concentrated in Europe, and largely absent from populations whose ancestors branched off earlier or migrated to completely different parts of the world. Researchers believe the mutation most likely arose after early humans had already settled into the European region sometime during the Ice Age. It is essentially a genetic fingerprint left behind by one specific ancestor from one specific time and place. And that fingerprint is still visible today in the DNA of their many descendants scattered across an entire continent and beyond. What makes this discovery even more interesting is what it reveals about how connected we actually are underneath the surface. Someone in modern Sweden, someone in modern Portugal, and someone whose family emigrated generations ago from Europe to the United States or Australia might never meet, might never know of each other's existence, and might look at their lives as completely separate stories. Yet if they carry this mutation, a lab test can trace a direct, unbroken line of inheritance connecting all of them back to one single person who lived on the same frozen landscape more than 20,000 years before anyone had ever built a permanent home. This is not a metaphor or a loose comparison. It is a literal, traceable biological fact, written into the actual molecules inside their cells. Every one of those carriers received their copy of this exact mutation from a parent, who received it from a grandparent, who received it from a great-grandparent, and so on, in an unbroken chain stretching back roughly 1,200 generations. Nobody in that chain chose to pass it on. Nobody in that chain even knew it existed for the vast majority of human history. It simply traveled forward automatically, copied and recopied every time a child was born, carried inside people who lived through the end of the Ice Age, through the rise of farming, through the birth and collapse of ancient empires, through plagues, wars, and migrations, all the way into hospitals and genetic testing labs in the 21st century. Modern medicine now takes this ancient inheritance very seriously because understanding it changes real medical decisions. People who carry the factor V Leiden mutation have a higher risk of developing dangerous blood clots, particularly in the deep veins of the legs, a condition known as deep vein thrombosis, and in the lungs, known as pulmonary embolism. Certain situations increase that risk even further, including long periods of immobility, such as long flights, certain surgeries, pregnancy, and the use of some hormonal medications like birth control pills that contain estrogen. Doctors now routinely consider genetic testing for this mutation when a patient develops a blood clot without an obvious cause, or when a patient has a strong family history of clotting problems. Knowing whether someone carries one copy or two copies of the mutation directly changes medical decisions, from how doctors manage pregnancy to what precautions are recommended before major surgery, to which medications might carry extra risk for that specific patient. A discovery about a single ancestor from the depths of the Ice Age is, right now, quietly shaping decisions being made in maternity wards and surgical planning rooms around the world. There is also an important point that this story reveals about how we think about genetic risk in general. It would be easy to hear about a mutation like this and think of it simply as bad luck, a flaw passed down through a family line. But the truth is more complicated and, in many ways, more interesting than that. This mutation was not born as a flaw. It was very likely born as a solution, a genetic adaptation that helped human beings survive one of the most dangerous moments in their biological life, the act of giving birth without any of the medical support we take for granted today. The reason it can look like a problem now is not because the mutation changed. The mutation is exactly the same today as it was 30,000 years ago. What changed is the environment around it. We no longer face the same constant threat of dying from bleeding during childbirth that our ancestors faced for almost the entire history of our species, thanks to modern medicine, safer deliveries, and access to emergency care. At the same time, we now face different risks that our ancestors rarely lived long enough to encounter, like long-haul flights, sedentary lifestyles, and certain modern medications, all of which can interact with this same mutation in ways that raise clotting risk. In other words, the mutation itself never became more dangerous. The world around it changed, and a trait that was once quietly protective became, under new conditions, a trait doctors need to watch carefully. This is one of the most important lessons hidden inside human genetics. Very few mutations are simply good or simply bad in every possible situation. Almost everything depends on context, on environment, on the specific challenges a person's ancestors were facing when that mutation first appeared and then spread. This same story plays out again and again across the human genome, in mutations linked to conditions ranging from certain blood disorders to immune responses to how efficiently different populations process particular foods. Traits that seem purely negative through a modern medical lens often turn out to have been genuine survival advantages at some earlier point in human history, shaped by specific pressures like disease, famine, extreme cold, high altitude, or, in this case, the dangers of childbirth long before modern medicine existed. What makes the Factor V Leiden story stand out is just how precisely scientists have been able to trace it. Most traits shaped by evolution developed gradually through many different mutations, building up slowly across enormous stretches of time, involving countless different ancestors, contributing small pieces to the final result. This is different. This is a single, identifiable genetic event, tied to a rough estimate of when it happened and a strong scientific theory for why it managed to spread as far as it did. It is about as close as modern science can get to pointing at a specific moment in deep prehistory and saying, here, this is where this particular thread of your biology begins, and that thread does not stop with Europe. Because of large-scale migration over the last several centuries, especially from Europe to North America, South America, Australia, and other parts of the world, this same mutation now appears far beyond the continent where it originated. Millions of people whose ancestors left Europe generations ago, and who may never think of themselves as connected to ancient European history in any meaningful way, are still, right now, carrying a direct genetic message from that one person who lived through the Ice Age. There is something genuinely humbling in realizing that a discovery this personal and this specific did not come from studying kings, empires, or famous historical figures. It came from studying ordinary blood from ordinary patients in an ordinary hospital, using tools that let scientists read patterns invisible to the naked eye. The person who originally carried this mutation was almost certainly not remarkable in any way that history would have recorded. They were not a leader, not a warrior, not someone whose name would ever have been written down, because writing itself did not exist yet where and when they lived. They were simply one person, living one life, in a small group of humans trying to survive a harsh and unforgiving world. And yet, because of one accidental change in their DNA, and because of pure chance combined with a genuine survival advantage, their genetic legacy did something that almost no other trace of their existence could do. It survived, physically unchanged, copied faithfully across more than 1,000 generations, and it is still here today, inside millions of living people, on multiple continents, still functioning exactly the way it did when it first appeared. This is really the deeper story behind almost every genetic discovery like this one. We tend to think of history as something written in books, carved into monuments, or preserved in ancient ruins. But an enormous amount of human history is also written directly into our bodies, inherited silently and automatically, passed from parent to child without anyone consciously deciding to preserve it. Every one of us is walking around carrying fragments of that history inside every single cell, whether we know it or not. The factor 5. Leiden mutation is just one thread in an almost infinite tapestry of inherited genetic history that exists inside every human being alive today. Some threads trace back to advantages against disease. Some trace back to adaptations for surviving extreme cold or high altitude or unusual diets. Some, like this one, trace back to the sheer biological danger of bringing new life into the world. Each thread tells its own story about what our ancestors faced and how their bodies adapted, sometimes in ways that only become fully understandable tens of thousands of years later, once science finally develops the tools needed to read them. So the next time you think about your own DNA or your own family history, it is worth remembering that your ancestry is not only made up of names on a family tree, old photographs, or stories passed down at family gatherings. It is also written, quite literally, inside your blood. In a language your body has been reading and copying correctly for tens of thousands of years, long before anyone ever thought to ask what it meant. Somewhere, in an icy landscape more than 20,000 years ago, one person survived, had children, and unknowingly started a genetic chain that is still unfolding today, in laboratories, in hospitals, and in the veins of millions of people who will likely never know that single ancestor's name, but who are, in a very real and measurable sense, still connected to them every single day.